Okay, so you are all welcome to our today's tutorial on acids, base, salt, and water. And of course, I can sorry for the problem we face on the internet, and I hope that will bring this is this will be the end of the network problem and. For the today's class, we are going to look at the concept of water, acids, base, and and salts. So let me share my slides. So I hope my slide is clear. So what is water? Water is the most abundant matter on earth, and it's also in all living creatures. And typically, organisms are constituted of about 70 to 90% of water. So you see, actually, we need to understand about the history of Earth and the component of Earth. So Earth is made up of water, land, and ice, and also rocks. Rocks, we have rocks, we have ice, we have water, and land. This is what constitutes to make an Earth. And of course, in addition, each of these components that make up these arts, they have their different percentage. And of course, the water molecule is the one that is the most abundant on earth. In fact, water occupies everything. So the largest part of the earth is water. So it's the most abundant on earth. And then second, water is very, very important. It is very, very important because it also found in a living organism. In fact, about 70 to 90% of each and every living organism is made up of water. So 70 to 80% of a living system is made up of water. So, but now the issue is, why is it that our body is made up of this huge percentage of water? Is because water must be present before metabolic activities can take place in a cell. And of course, in a cell, we have what you call metabolic activities. And what is metabolic activities? So whenever you had the word metabolic activities, it means metabolism. So what is metabolism? Metabolism is a sum total of all the biochemical reactions that are taking place in the body. Like for example, in human being or in generally in every living organisms, there are reactions that are taking place. And that what make up the activities that we do as a living organisms. Like as I'm talking now, like as I'm talking, is because of the reactions that are taking place in my body. And that is why I'm talking. And that is why also I'm talking the reasonable concept, and it's because of the chemical reactions or the biological reactions that are taking place in the body. And remember, ladies and gentlemen, in human or in human in general, brain is the master organ where all the activities of the body are controlled by the brain. So there is nothing that takes place in our body without the knowledge of our brain. So the brain will link with a lot of the proteins that are produced in our body to give us an instruction. Like for example, if we intend to talk or if we intend to see something, that message must be relayed to the brain. So once the brain receives that message, then a particular chemical reactions or biological reaction will take place in our body before we see, before our brain actually recognize what we see. So that is why we should understand that actually, like for example, let's take example of adrenaline. Let's take example of adrenaline. Adrenaline is a proteins as well as a hormone that control the fear and anxiety. It control fear and anxiety. So you see, adrenaline is not constantly there in our blood. It is only produced when it is needed. And it is degraded when the body doesn't need it. And it produced through the biological processes or the metabolic processes. So the synthesis of adrenaline is a hormone and protein at the same time. So it's a protein. So it means that it is made of by an amino acid. So when our body need adrenaline, the amino acid in our body will now come join one another, one, two, three, join another to join through a peptide bond to produce the adrenaline. And that is metabolism. So the processes whereby smaller molecules join together to produce a bigger one, like in this case, amino acid are the smaller molecules, while the adrenaline is the giant molecule. So the process in which smaller molecules join together to produce a larger molecule, that is what you call anabolism. So anabolism is types of metabolism. And we have catabolism. Like for example, once adrenaline is produced, we react to the situation. It's a fear and anxiety hormone. So when we are excited after producing adrenaline, then after some time we come back to our normal state. It's because adrenaline is being breaking down. So it's break down to produce the amino acid, and that is why we come back to our normal state. So there's a degradation and the synthesis of adrenaline at the same time. So this is just one of the examples of metabolic reactions that is taking place in our body. So that is metabolic activities. So we have thousands and the millions of these these metabolic activities that are taking place in our body. And of course, these reactions that are taking place in the body is what we call metabolism. So the sum total of all biochemical reactions, or the sum total of all biological, or the sum total of all reactions that are taking place in a living organism is what we call metabolism. So if there is no water, there is no metabolism. So no water, no metabolism. So it means that metabolism is only taking place when there is a water. And that is why sometimes we take drips. That is why we take drips, because sometimes if the amount of water, if the amount of water in our body is low, so what will happen? The metabolism will not take place, or even if it is going to take place, but it is going to be very slow. And that is why we develop disease conditions. So water is very, very important. And I hope, are you seeing the slide? Are you hearing me and also seeing my slides? If yes, please indicate. So that is one of the importance of water. And then second, water is referred to as a weak electrolyte. It referred to as weak electrolyte. So, ladies and gentlemen, I think at this point you should understand that water is the most abundant biomolecules that is found on Earth, and it is also the large component of living organisms, which about 70 to 90%. And it also referred to as weak electrolyte. So, what is a weak electrolyte? So, when we said weak electrolyte, it means that they are electrolytes that are partially they are partially dissociated. So when you say something is partially dissociated, it means that it is not completely dissociated to produce the component ions. So it means that when you said it is partial, it means that it is only some part of it. It is only some part of the ions that are dissociate, while some do not dissociate. Like for example, in water molecule we have H2O. But after the partial dissociation, we only have hydrogen ion and hydroxyl ion. So why is it that some hydrogen atoms still remain with oxygen in the hydroxyl ion? Because it is a partial dissociation. It's not a complete dissociation. So that is what you call that is water. So water is a weak electrolyte. Water is a weak electrolyte. So that is why when we say water is a weak electrolyte, it means that so when we said water is a weak electrolyte, it means that it is only partially dissociate. It is not completely dissociate. And that is why it's considered as a weak electrolyte. But when you said strong electrolyte, it means that it's a situation where the biomolecules or the molecules are completely dissociate. So when you have a molecules and those molecules are completely dissociate, so that described as a strong electrolytes. In fact, we even have non-electrolytes. We have non-electrolytes. So when you said non-electrolyte, it means that it is a situation where you have. Okay, let me just please share my slide again because I heard that some of you are complaining that they can't see my slides. So let me share the slide again. Okay, so let me share the slide again. So that is what you call a partial or a weak electrolyte. So I think in this slide you must clearly have, or you must actually go with three things: that water is the most abundant biomolecules on Earth, and it also occupies the largest percentage in living organisms with about 70 to 90%. And also, in addition, water is a weak electrolyte. And then we are also going to look at the concept of acid, base, and salt. So, what is an acid? An acid and base are defined with respect to their ability to gain or lose protons. So, whenever you have a substance and that substance loses a proton, that is, it releases a protons, it means that that substance is an acid. Assuming now you have H2SO4, and after you add that H2SO4 in water and you observe the release or the loss of protons, so it means that that substance is an acid. But when we realize that okay, you have a substance that after some time, then or after the reaction is taking place, then you have hydrogen added. It means that there is a gain of protons. So that substance is described as a base. Okay, then the next thing is after this is a buffer. Okay, before buffer, we need to define or we need to know the properties of water. So what are the properties of water? The properties of water constitute: one, water is the most predominant component of living organisms. And then also, yeah, okay, no problem. I'm coming. We are going to talk about acid and base very well. This is just the beginning. So water is the most predominant component of all living organisms. It is the most predominant. And of course, most chemical reactions in the cell are taking place in aqueous environment. That is where there is a water. And then hydrogen bonds hold the oxygen and hydrogen atoms together in a water molecule. So what does that mean? Generally, we have what you call a hydrogen bond in a chemical combination. So a bond that usually exists between hydrogen and the most electronegative element is called hydrogen bonding. So but now the question is oxygen electronegative element? The answer is yes. Oxygen, nitrogen, and fluorine are the examples, or are the typical example of electronegative element. So when you have oxygen and you have hydrogen joined together in a water molecules, so the bond that joined the two molecules together is called hydrogen bond. And of course, the oxygen that is present in the water molecule is electronegative element. And then the hydrogen is electropositive. So as a result of that, a water is a dipolar and exhibit a slight tendency to dissociate. So it is dipolar because it has negative component and then the positive component. So the negative component of water is oxygen and the positive component of water is hydrogen. So that is why they can actually interact, positive and negative. So it is dipolar. So as a result of that dipolar nature, it is slight, or it has a slight tendency to dissociate. So that slight tendency of dissociation is what we describe as is what we describe as partial dissociation, as we discussed from the previous slides. And then the next thing after this is, of course, the biological importance of water. So, what are the biological importance of water? The biological importance of water include water molecules with electrical charge distributed unequally about its structure is referred to as a dipole, because of course it has negative and negative and positive, so it is unequally because they are opposite charges, and that is why it is called a dipole. So generally, when water molecule dissociate, it produce hydrogen ion and hydroxyl ion. So the strong dipole and high dielectric constant of water is what enables the water to dissociate large quantities of charged compound. So that is why, remember, ladies and gentlemen, water is considered as a universal solvent. So why is it that the water is considered as a universal solvent? Is because especially where you have a charged compound, like the compound that are formed, like the compound that are formed by electrovalent bonding. So generally, the compound that are formed by electrovalent bonding are easily dissociated in water. And why? Is because of the strong dipole and the high dielectric constant. So, ladies and gentlemen, please, as we are discussing this, you should know whenever you had the word water as a universal solvent, is because the water is is a strong dipole and will also have strong or high dielectric constant. and that is what enable water to dissociate a large quantities of a charged compound. So that is why water is called universal solvent. And then in addition, the presence of hydrogen bond also enables water to dissolve many organic molecules that contain functional groups. Yes. Like for example, remember in organic chemistry is divided into two. We have hydrocarbon and we have non-hydrocarbon. So in the case where we have non-hydrocarbon, most of those non-hydrocarbon they have a special functional group. Special functional groups. And what is a functional group? A functional group from the basic knowledge of organic chemistry, it can be an atom, it can be a group of atoms, it can be a bond. That is either single bond, double bond, or triple bond. So it can be atoms, group of atoms, or bonds that usually determine the chemical properties of a compound. So it determines the chemical properties of a compound that describe functional groups. So because of the presence of hydrogen bonding that is there in water, that is why a lot of organic compounds that have a functional group, they also associate there. So ladies and gentlemen, I think you can clearly understand the three major reasons why water is considered as a universal solvent. Is because of strong dipole, one, high dielectric constant, and then the presence of hydrogen bond. So these are the three things that make water as a universal solvent. And also, water provides the enabling environment for macromolecules to achieve stable structure in a solution. I hope you can all hear me because I saw that some people are complaining. So please, those that are listening and seeing my slide, please indicate by saying yes so that let me know that the problem is not from my side. So generally, there are a lot of macromolecules. An example of macromolecules, ladies and gentlemen, remember we have actually started introducing ourselves to different classes of macromolecules. And these classes of macromolecules, we have carbohydrates, apart from carbohydrates, we have proteins, we have nucleic acid. All these are macromolecules. So what make the macromolecules to be stable, to have their stable structure in a solution? So that at least their three-dimensional structure or their structure will not be or are not distorted. So the structure of macromolecules are achieved due to the enabling environment provided by the water. So water provide an enabling environment that make the stable structure of the macromolecules. And let me tell you, ladies and gentlemen, if the stable structure of the macromolecule is not being maintained, it means that there must be a problems because there are some biomolecules that the only functions, in fact, not some, almost all biomolecules or almost all macromolecules that are found in living organisms, they maintain their functions in their stable structure. And once they lose their stable structure, once they lose their stable structure, so they will not function, and that is the end of that is the end of it. Like for example, especially enzyme function when they are in their stable structure. And these enzymes, a lot of enzymes, they are what even make life possible. Because if there are no enzymes, there will be no reactions. And if there is no reactions, of course there is no life. Because what makes cells function is because of the reaction. And if there is no cell, so no life. So no reaction, no cell, and no cell, no life. If there is any problem with the stable structure of any macromolecules in your body, there will be a disease. In fact, ladies and gentlemen, let me say this because you are nurses. Why are you doing biochemistry? Is because you are studying macromolecules. And what are these macromolecules? They are the molecules of life. So most of the diseases that we are coming up with, or most of the diseases that patients are coming up with, they are associated with these macromolecules. In fact, almost every disease that you know, it has its own biochemical basis. If you take diabetes, it's because of the unstable structure of the carbohydrates, because it's the glucose. Because of course the concentration of our glucose in the blood should be within 4 to 5 milligram per deciliter. So because the unstable or the stable structure of macromolecules is distorted, which is the glycogen, so that is why a lot of concentration of the glucose will be there in the blood. And as a result of that, hyperglycemia and then glucose. So that is why we should understand that water is very, very important. And that is why sometimes if there is no water in the body system, why is it that if you are given drip, is it not a normal saline? It's a normal saline. That is 0.9% sodium chloride. So why is it that sometimes if you have a patient and you check that the water in that body system, once you observe that there is a low IQ's environment in that particular patient, you know that there is a problem and you give the patient drips. It's because the percentage of water is low. And if the percentage of water is low, so many organic compounds will not be dissolved and you want them to dissolve. So many charged ions or so many charged compounds, like remember you are doing physiology and anatomy. Potassium and sodium, calcium ions, they are very, very important in the body. So if they are not dissolving what will happen? There will be a problem. And so also at the same time, the stable structure of the biomolecules, or the stable structure of the macromolecules. And if there is a problem on those macromolecules, of course, there's still a problem. There is a huge problem. So water is very, very important in the body system. It's very, very important. So you should understand, at least from this slide, you will understand that because of the strong dipole and high dielectric constant, that is why water was able to dissolve the large molecules of a charged compound. And the presence of hydrogen bond also make it to also dissolve a large amount of organic molecules because of the functional groups. And it also maintains the stable structure of macromolecules to achieve their functions. So this is actually the importance of water. So you should understand that water is very, very important, and that is the major reason why you need to check whenever there is a problem. Whenever there is a problem, or when the percentage of water in the body is low, so you need to keep the patient drip. And why is it that you are giving the normal saline? It's because to maintain the osmolarity. Okay, so the next thing is actually let's look at another slide. So now, what is an acid? That is the next thing that we are going to look at. What is an acid? An acid An acid are substances that break down to release hydrogen ion when dissolved in water. So generally acids, when you dissolve them in water, they release hydrogen ion. So anything that you know, anything that you know that after adding it in water, it will release hydrogen ion or it will produce hydrogen ion. So that substance is considered as an acid. Like for example now, look at it. This is a substance and you're adding it here, and after adding it in water, there are like five ionic form of hydrogen ion. So that is an acid. Although from the basic knowledge of our chemistry, even down to SS1, we have what you call Arrhenius definition of an acid, Lewis definition of an acid, and Bronsted-Lowry definition of an acid. And each of these scientists have their own definition. They define acids on different bases. Like for example, Arrhenius definition of an acid. This definition that we shown here in this slide is an Arrhenius definition of an acid because it's the person that define acid as a substance that produce hydrogen ion in a solution. And then apart from that, we also have Lewis. That is, an acid is a substance that accept a lone pair of electrons. An acid is a substance that accept a lone pair of electrons. So there are these different definition of an acid, but here you should understand that whenever you have a substance and it releases hydrogen ion, then that substance is described as an acid. So when the hydrogen ion are released, the new solution is set to become acidic. So because the hydrogen ion are released into the solution or into the new solution, so that is described as an acidic. So it now changed to acidic. And that is why, ladies and gentlemen, remember from the basic knowledge of your physiology and even from the biochemistry, we have what we call metabolic acidosis and we have metabolic alkalosis. Because each and every environment in our body, has its own specific pH. And once that pH is altered, it means that the physiological processes in that particular environment will also be affected. It will be affected. Like for example, if you have, let's say in the mouth, generally the pH of the mouth is around 6.4. Yes. So now assuming now you will take anything that is acidic, what will happen? The hydrogen ion will release there. So if the hydrogen ion are released there, what happened? The pH, because it is somehow neutral and you are now releasing hydrogen ion. So acidic substances usually have a pH lower than 7 because we have what we call a pH scale. So when an acid substance is released, let's say in your mouth, of course the pH of your mouth will be altered. It will be altered. So in any situations where an hydrogen ion is released or where a hydrogen ion is released, the pH of that environment is affected. So if the pH is not maintained within the normal physiological pH, so there will be a problem. And that is what we call metabolic acidosis. The pH of the stomach is 7 point, sorry, of blood is 7.4. So if you now take any acidic substances, or if your pH in the blood is being affected by dropping an acidic substances, what will happen? The pH will go down from 7.4 to maybe somehow 5 point something. So it means that the pH is reduced. And that condition is called metabolic acidosis, or is called acidosis. And then the next thing after this, generally, an acidic substances or acids might dissociate partially or completely. And acid substances can be partially dissociate or completely dissociate. Like for example, acids acids is classified on different bases. We can have organic acid, we have inorganic acid. So that is based on the source. We classified acid based on their sources. Like for example, we have what we call organic acid and inorganic acid. So when we say organic acid, it means that they are acids that are obtained from living organism. So they are acids that are obtained from living. That is why it's called organic. For example, citric acid is from orange and it's an organic acid. Garlic acid is an organic acid. And then after that, we also have inorganic acids. And when you say inorganic acid, it means that it is coming from inorganic substances. That is inorganic acids. You are nurses, so sometimes you need to understand the situation of your patient. It's not always that you put your patient on drugs or on injection. That is why you are doing biochemistry and nutrition, because that will give you an idea on how to manage some of the diseases. So it's not in every cases that will just start giving patient drugs. Like for example, now assuming you have a patient, let's say a patient with ulcer, will you advise that particular patient to start taking orange? I'm waiting for your response. Please let someone respond on this. Is it possible to ask your patient take orange? I'm waiting for your response. I am waiting for your response. Is it possible to advise your patient having ulcer to take orange? The answer is no. Because ulcer is an acidic disease. It's an acidic environment. It is actually acidic. Because of course, remember, gastric juice is an acidic environment. So you can't advise your patient to start taking acidic substances. So the best thing, if you want, that is why most of the anti-cancer drugs, they are called antacids. So if you have a patient, I think advisably the best thing that you are going to do, you advise your patient to start taking an alkaline foods like fruits. You cannot advise your patient to take orange, but you can advise your patient to take something like apple, because apple is an alkaline food, or is an alkaline fruit. So it will neutralize the acidic in the stomach. Acids base neutralization. So you'll advise your patient to take alkaline food. So that is why, as a nurse, if you have a patient suffering from ulcer, it is always advisable. It is always advisable to tell or to put your patient on some actually uh, on some foods. So it's not always giving patient drugs, because even drugs in the body is a dangerous. It's a dangerous substance. So we don't always encourage people to be taking drugs. So it's just control diet. You can control their diet. So then the next thing is actually we have, apart from the organic acid and inorganic acid, we also define acid based on their dissociation. So we have weak acid and we have strong acid. So a weak acid is an acid that is partially dissociated in water. So it is partially dissociated in water. That is what you call, that is a weak acid. And we have a strong acid. A strong acid are acids that are completely dissociated in water to produce hydrogen ion. And of course, in addition, ladies and gentlemen, we should understand that in a solution, weak acid establish equilibrium between the proton and its conjugate base. Between a proton and a conjugate base. So what is a conjugate base? Slide here, we can see that it is He plus water, it dissociates to produce hydrogen ion plus A minus plus minus. So the A minus here is the conjugate base. And of course, you have your proton. So usually an equilibrium position is maintained. So that equilibrium position is always written. So the equation represents the dissociation of He into hydrogen ion and A minus. So applying the law of mass action. That is, how do you apply the law of mass action? Is by taking this kind of bracket, this bracket in chemistry, you call it concentration. This kind of bracket. So we have Ka. So we now take the concentration of the products. So what is the concentration of the products? The product is hydrogen ion and A minus. So hydrogen ion concentration in bracket times iodide times A minus in bracket divided by the concentration of Ha. Divided by the concentration of Ha. So where the Ka here. So but now the question is, someone will ask, why is it that? Because here we have Ha plus H2O on the reactant side, and then of course in the product we have hydrogen ion and A minus. So someone will ask, where is hydrogen in this expression? Because we only have hydrogen ion concentration, A minus concentration divided by the Ha concentration. Where is water? So generally, the concentration of Ha, the concentration of Ha, sorry, the concentration of water is usually normalized. So we don't express it. And then we have a base. What is a base? A base is a compound that accepts a proton in aqueous environment. So it's a substance. So in this case, A base is a substances or are substances that accept a proton. So in this case, they are accepting proton. So we have a calculation. Okay, but before then, we have what you call a pH scale. Or before we talk about the pH scale, we have what you call indicator. What is indicator? An indicator is any substances or any material that can detect whether the sample or the solution you are giving is an acid or base. So that is what you call an indicator. So an indicator is a substances or material that can detect whether a solution is an acid or a base. So we have different types of indicator. We have methyl orange, we have phenolphthalein, we have litmus paper. Like for example, please, I hope you can see my slide. I saw that some are complaining. Maybe they are just coming. So please, are you seeing my slides? So what I'm saying now is that we have okay. Okay. So let me share my slide again. So what I am saying here is that so as we are talking about uh, an acid, sorry, a base is any substances that accept a proton. So a base usually accept a proton. Yes, I'm actually trying to share the slide again because some of you are complaining that they can't see the slide. So I'm going to share it again. Yes, I'm coming. I'm trying to share the slide again. So now I was able to share it again. So that is an acid. So as we are talking about an indicator, what is an indicator? So an indicator is any substances or material that can be used to test whether the substance is an acid or a base. So we have litmus paper. A litmus paper, for example, if you have a red litmus paper, and you put it inside a substance or inside a sample. So if that litmus paper change from red to blue, so it means that that solution is a base. And of course, if you have a blue litmus paper and you put it inside a solution and it change from blue to red, So that tells you that the substance is a base. It's like some of you are still complaining that they cannot see the slide. Please, can you see the slides? Is anybody seeing my slides? Please respond. Is anybody seeing my slides? Okay. So let me try to share it again. So I hope my slide can clearly, I hope now you can see the slides. So if you can see my slides, please indicate by saying yes. Can you see my slides now? Dinat Johanna, can you see my slides? Feds, can you see my slide now? Oh, still not showing. Okay. Okay, so I'm now going to share it again. So I hope now you can see the slides. Can you see the slide? So can you see the slide now? So that is an indicator. So we have so many of them, phenolphthalein, orange, and we have so many of them. So we have what you call a pH. What is a pH? A pH can be defined. A pH can be defined as a negative logarithm of hydrogen ion concentration. Are you sure you are not seeing my slides? So a pH can be defined as a negative logarithm of hydrogen ion concentration. And that is where we have pH is equal to minus log of hydrogen ion concentration. Okay, Dinata, are you seeing my slide? Dinata Joshua. Oh, sorry, Dinata Johanna. It's still not showing. Sakina Musa. My slide is still not showing. Please respond. So, so that is actually what we call okay, let me try and see again. So we have we have what you call a pH. So a pH is a negative logarithm of hydrogen ion concentration, and we have formula for calculating pH. So the formula for calculating pH is equal to minus log of hydrogen ion concentration. And we also have a formula for calculating OH, that is POH. So POH, wherever you see H indicate an acid, while OH indicates a base. So you can calculate pH and you can calculate POH. So POH is minus log of OH, while pH is log of or minus log of hydrogen ion. So we have a pH scale. And what is that pH scale? So let's now look at pH scale. So I hope you can see these slides. This is another slide. I hope you can see this one. Now I'm sharing another slide. Can you see this one? So we can have, or we can calculate pH and we can calculate POH. So how do we calculate pH? So pH is minus log of hydrogen ion concentration, and we have POH is minus logarithm of OH concentration. So I want us to understand something very, very important. We have a pH scale. So when you have a pH between 0 to 7, that one is an acidic. And the lower the pH, the higher the acidic. The lower the pH, the higher the acidity. So then we also have, apart from 7 to 0, we have also 7 to 14. So when you have a pH above 7, so it means that it is an alkaline medium or it is an alkaline environment. It is an alkaline environment. or is an alkaline medium, or is an alkaline solution. So, but when pH is 7, when pH is 7, so that is a neutral pH. So we should understand something. Most of the water that we are taking, it should be within a pH of 7. So that is why sometimes it is advisable, or you are always advisable to be taking. It is always advisable to be taking. It is always advisable to take a water that is, of course, within or with a pH of 7. And most of the water that we are taking is not a neutral water. It is not a neutral water. So that is why we have to be very serious with the types of water that we are going to that we are taking. Actually, there was a time, I think it was actually when I'm doing my IT, that is service, around 2014, in 2014, 2014, 2015. We collected uh, we collected water from different hostels in ABU. So we use a pH meter. We have a pH meter. So a pH meter is a meter that will give you an idea of what you are doing, or what you are measuring, whether it is an acid, base, or is a neutral. So we collected water from ABU environment here. Surprisingly, most of the water that we measured within a pH meter, all of them, they are within acidic. They are acidic water, and now we are taking them. We are taking acidic water, and that will contribute with a lot of medical conditions. And we are taking the water constantly all the time. So that is that problem. So we should understand that a pH, a pH of each and everything that we are taking, we have to be serious of what we are taking. Because sometimes a problem of taking food that are acidic or basic can lead to a lot of medical conditions. So, ladies and gentlemen, I think from this explanation, you will understand that understanding pH is very, very important. And let me tell you again. Do you know that sometimes, because of lack of our knowledge of understanding the acidic and the non-acidic environment, can lead to a lot of problems? Like, for example, if you are farming, even a soil, we have different types of soil. We have acidic soil, we have basic soil, that is alkaline soil, and we have neutral soil. And so many of us sometimes will just go and plant our seeds in an environment where it is acidic. An acidic environment usually doesn't support the growth of most of our food products. So that's where sometimes we usually have a low yield. We get low yield. So you have to be very serious about that. We have to be very serious about what we are taking and the pH of what we are taking. So because of the problem that some of you are complaining, or you are complaining that you are not seeing my slide, I think it's actually a network issue. So in the next class, I want all of you to follow all this video from the beginning to the end because the explanation is there and is very clear. So just watch the video from beginning to the end, and on Saturday, this is where we are going to continue with. We are going to talk about the pH, how to calculate pH, how to calculate pOH. Yes. So ladies and gentlemen, if there is any question, you can ask question before we close for today. So I'm waiting for your questions. Is there any question? So if there is no any question, I think we are going to stop here. Thank you.